Our speakers today are Adrian Benjamin, Senior Commercial Development Manager of Oncology and Cancer Research of Illumina. Adrian Benjamin is a Senior Commercial Development Manager for Oncology and Cancer Research at Illumina. He focuses on genomic applications in oncology, working with internal partners and customers to meet the needs of this dynamic and complex market. Anup Madan, PhD, Senior Technical Director, Genomics Services, Covance. Dr. Anup Madan leads a team of experienced genomic scientists that support multiple studies within the drug development process. In addition to managing laboratory operations, his team is responsible for developing novel assays, analysis of high-throughput genomic datasets for identification of biomarkers for patient stratification and transitioning these into clinically relevant diagnostic assays. Stephen Anderson, PhD, Chief Scientific Officer of Covance. Dr. Stephen Anderson is the Chief Scientific Officer of Covance. In the CSO role, he leads the development and implementation of scientific strategy throughout the various Covance business units, coordinating with the scientific teams in early development, central labs, clinical development, market access, and the LabCorp diagnostics business. Okay, now let's begin. Adrian, please go ahead.
Thanks so much, Michael, and I apologize for a little bit of delay in getting started. I'm really just going to start here by overviewing the agenda for everybody and then passing things over to our first speaker. So as Stephen Anderson is going to cover sort of an overview of immunotherapy and the importance of biomarkers, we're going to move over to Anup, where he will cover and go much more in depth, focusing on leveraging advanced genomic techniques in biomarker discovery. And then I'll end with some quick closing remarks, highlighting a review paper that I think sort of brings home a lot of the things that will be on this webinar, and then hopefully have some time for some Q&A. So with that being said, I am going to pass things over to Steve to start us off. And I will give you the ball, Steve.
All right. Thanks, Adrian, and thanks everybody for joining today. As Adrian mentioned, I am going to provide a very brief overview of the importance of immuno-oncology and immunotherapeutic approaches in cancer treatment. In addition to that, I'll talk a little bit about biomarkers that are in current use today, as well as those that are being evaluated for discovery purposes and to move into trials and eventually for patient diagnostic purposes. And I'll finish up with giving a very brief introduction to genomic biomarkers that are relevant, and then turn it to my colleague Anup Madan, who will really focus on the genomic biomarkers and the value of next-generation sequencing as a tool to look at the immune system, to look at features of the tumor and how those are relevant in this whole area of immuno-oncology. My first slide is just a cartoon representation of the role and the various ways in which the immune system can be activated to attack cancer cells. There are a variety of approaches in immunotherapy. We've heard a lot and seen a lot of great progress looking at checkpoint inhibition, looking at the checkpoint blockade axis with the relevance of molecules like PD-1 and PD-L1. There are also activators of the immune system that are relevant, so overcoming the brakes on the immune system, increasing the activity of the immune system. There are ways in which an individual's T cells can be engineered through technologies like CAR-T to attack the individual's tumor cells. And then also cancer vaccines, which have been an area of interest for several decades, but now with the increased understanding of the immune system, also show additional promise as we move forward. As I mentioned, the checkpoint blockade and inhibitors of that blockade have been a relevant target for therapeutic development. So we have receptors on T cells, we have ligands on tumor cells that interact to suppress the immune system's ability to attack tumor cells and therefore provide a mechanism by which tumor cells evade that activity. Agents that interfere with that interaction, like monoclonal antibodies against PD-1 or PD-L1, remove that suppression and reactivate the immune system. Currently, there are 4 approved drugs, and these include drugs to CTLA-4, PD-1 or PD-L1 that address checkpoint inhibition, and there are a variety of indications for which those therapies are currently approved and in use. Melanoma, bladder cancer, renal cell cancer, head and neck cancers, certain types of lymphoma, and non-small cell lung cancer. So an area of great interest and an area of great success. While the checkpoint inhibition molecules have been a primary focus, this slide just points out that that's the beginning of how we look at addressing immune therapies in oncology. We're looking at the capability to move from single agents to combinations of agents, as shown in this slide. It could be a combination of 2 checkpoint molecules. It could be a combination of an immuno-oncology molecule plus traditional chemotherapy or targeted therapy, or it could be a combination of an inhibitor and an activator of the immune system. So there's a tremendous amount of interest in how those different strategies can be synergistically and provide even more impressive results than just that have been seen with attacking the checkpoint blockade. When we look at and consider the biomarkers that could be relevant for understanding the interaction of the immune system and the tumor, there are a variety of technologies and analytes that we could evaluate. So on the lower left part of this, proteomics, so looking at the expression of the receptor, program death1, or the ligand PD-L1 on tumor cells, phenotypic characterization of the infiltrating lymphocytes, etc., are already in use. On the genomic side, mutational burden analysis, mutations within polymerases or repair genes that create increased mutational burden or genomic instability. Anoop will talk a lot about those tools, as well as understanding the expressed genes within the tumor. Epigenomics, looking at other ways in which genes are either activated or repressed. And then included in this would also be immune function genes, so looking at, again, the response of the immune system to the tumor. And lastly, included in this slide is even the impact of the microbiome or previous exposure to viruses that may be relevant to priming the immune system in such a way that it can be manipulated and enhanced in its attack of tumor cells. So as laboratory scientists, we always like to have all of the appropriate tools in our toolbox to look at biomarkers. And this slide just shows that biomarkers can be cell-based, so technologies that look at T and B cell subsets, as shown in the middle with the flow cytometry histogram, the expression of chemokines and cytokines, as is shown in the upper left-hand panel. Tissue-based markers, again, expression of program death ligand 1 in tumor cells, as is shown in the upper right quadrant. And then lastly, genomic biomarkers, mutational burden, the production of novel or neoantigens, markers that are hallmarks of genomic or genetic instability, analysis of the clonality of T cells, etc. All are tools that are relevant in the toolbox as we look at identifying the appropriate biomarkers that are associated with potential response to immunotherapeutic approaches. Currently, the assay, an assay that has been stressed in evaluating the checkpoint inhibitors is the expression of PD-L1, the ligand on tumor cells. This is shown in the left-hand box where we are looking at multiple tumors, low magnification, high magnification, expression of the ligand on tumor cells, going from the left-hand side of no expression to high level of expression on the right-hand side. In the Kaplan-Meier curve, and this is from data looking at response rates and PD-L1 expression levels for Keytruda or pembrolizumab, showing that patients that have an expression level of greater than 50% respond very well to treatment, and lower levels of expression, lesser levels of response. This has led to the development and commercialization of companion diagnostics. Those assays required to use a particular therapy, like the DACO22C3 assay and Pembro, and complementary diagnostics, where the assay information provides additional information for the clinician around the benefit of using the therapy or potential risk, but it's not required. The next slide just shows the challenge that exists today with regard to some of the lead molecules being developed around anti-PD-1 or anti-PD-L1. And if you go down about halfway through the chart, what you'll see is that these four major drug developers looking at this blockade area have partnered with different diagnostic manufacturers, Dako, Ventana. They've chosen different antibodies that are used to detect the PD-L1 ligand. They look at potentially different components of the tissue, tumor cells versus sometimes including infiltrating lymphocytes. And most importantly, they look at different cutoffs, what's a positive or what's a negative. And so this has caused some confusion or some challenges, in particular as these assays and the drugs become available commercially, to how clinicians would evaluate and use the various technologies necessary for use of the therapies. So those markers are currently in use. There are a variety of genomic biomarkers that are relevant and have been shown to be relevant in trials, with a continued evolving landscape here. And I'll point out just a few things. I've mentioned them briefly, and then I'll turn it over to Anoop that'll go into much greater detail. Mutational burden or load appears to be relevant in some tumor types like melanoma and non-small cell lung cancer, where there's an environmental component that increases mutational burden. Increased mutational burden is often associated with then an increased likelihood of producing a protein that is novel with regard to its recognition by the immune system, a neoantigen. We know in colorectal cancer and other potential tumor types that genomic instability, like microsatellite unstable tumors in colorectal cancer, have a different response rate than those where there is a genomically stable tumor. I mentioned briefly the T-cell repertoire. Anoop will talk a little bit about this. And then gene expression profiling, as we look at phenotypic and genotypic characteristics of both the tumor and also of the immune system responding to the tumor. And with that introduction, I will turn it over to Anoop now, who will go through some examples of how NGS technology can be applied to look at these various genomic biomarkers in immuno-oncology. So Adrian, if you could pass it on to Anoop, that would be fantastic.
Yeah, I'm trying to do so. If somebody from Fierce can do that, the method that I was shown on how to do that, was unable to do it. Thank you.
Yeah, I got it. Thanks, Steve, for setting the stage for next phase of the talk, where I will focus mostly on various genomic assays, mostly sequencing, that we are using not only to stratify patients, but also to identify if other genomic aberrations can be used for immunotherapeutics and advance the field. I will also focus in the end on how we are using these assays to identify markers that can be used to monitor patients who respond to immunotherapies or not, which are of importance both from clinical trials perspective as well as the diagnostics. The use of immunotherapy as one of the pillars to treat patients with metastatic tumors gained prominence somewhere around in the middle of 2015 with series of landmark papers that were published in Science. As Steve mentioned, these articles showed that patients with a higher mutational burden load respond better to the immunotherapeutics. Those of you who are following the field, there have been increased in number of publications ever since, where primary focus has now shifted to define the expressed neoantigens using both RNA-Seq and exome-Seq data. What we can do with the exome-Seq is define the expression of non-synonymous mutations using patients' tumor samples. And then RNA-Seq is being used to determine which of these variants are expressed. The option of immunotherapy has given hope to patients with metastatic cancers and is being used to treat patients of practically every tumor type. And every day we hear stories that instead of going to hospice, a person is getting the benefits of immunotherapy and leading a normal life. In fact, today the first thing patients and their family members when they walk into the clinic ask, am I going to get the immunotherapy? Will it be effective? And is my tumor will be genetically defined? This is a paradigm shift from 3 to 4 years as we understand more and more. It's going to revolutionize the patient treatments. And on scientific front too, it has changed how we use various technologies. Few years back, we used to do RNA-Seq or microarrays or other hybridization-based approaches because of its dynamic range and better sensitivity. What we used to sell is that we can use RNA-Seq not only to detect genes which are expressed at a very low level, those are practically hidden in the noise of the microarray. You can keep on sequencing and you can detect more and more. The price of sequencing has gone down significantly. I still remember 12 years back, we paid about $100,000 to just sequence 3 samples and generated 35 base pair length, 1 million reads to get better resolution of genes in GBM. But apart from pricing, today the question is not about sensitivity and dynamic range anymore. The question we have to ask is what information we can derive from data sets that cannot be obtained for hybridization-based approaches. So I told earlier that one can look variants from both exome and RNA-Seq data. But still there are computational problems which inhibit to use such an approach. And that largely derives because of the filtering effects due to low quality scores, or in some cases the variant is expressed at a very low level in RNA-Seq data. If the genes are expressed really at a low level, 1 copy or 5 copies, it is very, very difficult to identify the variants from that data set. And as a result, a lot of investigators ask me, can we just use RNA-Seq data? We are looking into mutatopes which are expressed. So why do we need to do DNA and RNA-Seq together? And I always tell them you need to better understand what genes are expressed and what mutations are accurately defined. You need to sequence both RNA and exome data sets. So at Covance, we have developed the tools by which you can combine both these data sets and remove some of these artifacts to better define which of these variants are expressed. We can go down to a single copy genes where some of these variants are expressed without any problems, or genes which are expressed at 3 or 4 copies as shown in the right-hand side of the graph of this picture. These tools also align the indels properly so that we can easily identify which of these variants are expressed properly or not. In addition, we also looked into RNA editing effects. These are the novel class of variants which are not present in DNA, but are the noise of the system, RNA editing, but these can be used for Neo-epitope predictions and all those things. So in order to test these tools, I will talk about this, come back to this slide a little bit later. We collaborated with Gibbs Cancer Center, where we analyzed about 60 tumor samples with different RAS signature scores. This data set was generated with the perspective of defining technologies that can be used for patient stratification using the RAS signature score also. So while the field of immunotherapy was exploring, we asked this question, like, can we use this data set and see which Neo-epitopes are present, and if any of these patients could have benefited from the immunotherapy or not, following those science articles that we just talked about. And we collaborated in experiments with MedGenome, a small company, where we wanted to look into how many of these mutations are expressed mutations and are present in both RNS-Seq and exome-Seq data. And we identified in this whole subset there are about 650 or so mutations. Then we generated this random peptides from that 9 peptides using the proprietary tool, how many of these peptides are present, and we were able to identify about 6,000 peptides or so. And then we used the HLA binding predictions and other tools and to identify which of these peptides can be prioritized based upon these algorithms that can be taken into the clinic or that can be used for patient identification. Now this is a very low number. Normally you see a very high number, so we were first a little bit doubtful that are these approaches working or not. In order to see that, what we did was looked into the TCGA publicly available data sets, and in that case also in colorectal cancers, the number of tumor Neo-epitope burdens was very low. And other thing which we also identified that these are mostly personalized to individual tumor type, which sets the stage for personalized medicine also. So then we started looking into the gene expression signature, and when we looked into it, we identified that there are myeloid-derived suppressor cells, which are largely expressed in this population of cells, and there are two different categories these tumors can be stratified, one with a high MDS score and one with a low MDS scores. And these were basically done by looking at the immune cell signatures, which were characterized to look into CD4, CD8 T cells positive, macrophages, dendritic cells, and natural killer cells. And again, the same pattern was also observed in the TCGA data sets, where you can classify into a high MDS as well as the low MDS cells. So we then wanted to look into this a little bit more, that how does these correlate with the RAS signature score. Let me spend 2 minutes on this RAS signature score that was developed a few years back, about 10 years back, as a part of Merck, where we looked into various tumor types and identified expression level of 16 or so genes which can stratify the colorectal cancers. Later on, this scores was refined by AstraZeneca, and then they looked into which other genes can be added on to that or not. The whole purpose of doing this exercise was that it takes some point of time to analyze your RNS-Seq data and the DNA-Seq data too. And sometimes the patients don't have that much time. You want to look into these biomarkers and see which one is a likely candidate for immunotherapy and which one is not likely candidate to immunotherapy. And some of these approaches can be done in a relatively faster turnaround time, just to predict whether you are going to respond to immunotherapy or not. So we have this grant from NCI where we are looking at these tumors to see how do we define these technologies, which technology is the right technology to do these assays. Recently we published 2 papers. I'm not going to go too much into details into that. You can look at all this online, where we have used various technologies to see how to transition a biomarker from a research lab into the clinic for clinical trials. So since we have this data earlier, I asked questions that people are asking, like how do you use these data sets, not only to stratify patients, but what else you can explore from there. One of the things which is missing from the data sets is the use of gene fusion events as well as the splice variants of it. Can these be used for neoantigens or not? These are novel neo-epitopes that are expressed only in the tumor cells, and currently nobody is using them. So what we thought was, let's look at these events and see, do they correlate with the RAS signature scores or not? And as shown in this slide, we were able to predict about 300 or 400 gene fusion events present in about 60 tumor samples, and their expression level was also correlated with your RAS signature scores. And be used not only for prediction for patient stratification, but it also gives you an idea that these novel new epitopes are present that can be used for immunotherapy or not. Some of these things you cannot find in the hybridization-based approaches and are unique, and that can be identified only from sequencing data sets. So we still have a long way to go as the field of immunotherapy is evolving. As Steve mentioned, there are multiple approaches. We need to understand better which of these approaches can be used. So as a part of LabCorp Covance, what we are doing is using other approaches to better understand which of the biological pathways play a role in tumor biology and how we can exploit them in the field of immunotherapeutics. We are doing methylation analysis to better understand how the methylation levels can be exploited, in addition to the ChIP-seq and the RIP-seq. Both of these are important for determining epigenetically regulated gene sets. Then we are using various tools like cell-free DNA and T-cell, B-cell repertoire sequencing to monitor the response in the patients with the immunotherapy approaches. And I will show you a few examples of those in the coming slides. So this is a lovely paper which came recently in Clinical Research, where these investigators showed that if you treat these patients with the epigenetic drugs, you can enhance the expression level of the genes which are present on the surface of the tumors and exploit them from immunotherapy. It's a huge area of interest for me. I published a few years back, as I mentioned, we spend a lot of money in determining which of the genes are expressed and are epigenetically regulated in glioblastoma multiforme. While we were doing these studies, we identified a lot of the tumor proteins encoded by cancer germline genes, such as MAGE family of genes. These genes are epigenetically regulated, and you can increase their expression level by both DNA methyltransferase inhibitor as well as the HTAC inhibition. At the same time, we had also generated the whole genome sequencing data on these patients and the whole exome sequencing. And RNS-Seq was recently done as a part of Covance. Now what we are trying to do is look into these data sets and see which of these expressed genes are mutated and whose expression is epigenetically regulated. And the whole idea is, by giving your DNA methyltransferase inhibitor, can you increase, as I said, expression level of these genes so that they can represent a novel battery of neo-epitopes that can be targeted for immunotherapy. And we are doing the similar thing in the breast cancer, where we have looked into these patients to identify which of the genes are epigenetically regulated. And I said that there are certain aspects which you can only look into by sequencing-based approaches. These things are hidden. This is the MAGE family of proteins, which are very, very similar in their sequence. If you look into the hybridization-based approaches, one cannot identify the expression level of these genes very accurately. We have done this experiment with nanostring, we have done this experiment with qPCR and other approaches, and everything looks good if you are looking for just expression of these genes. But when you probe closely, by looking at very small base pair differences, we can look into RNS-Seq data and more accurately define which of these genes are expressed, at what level they are expressed, so that they can be used for targeted therapeutics. The other thing which also RNS-Seq data can show, in certain cases you are looking at the gene expression profiles, you know that it is expressed, but your antibodies don't bind. And when we closely look at some of these genes, you can see if frame shift mutations are present. As a result, the targeted protein is not produced, and that inhibits the antibody binding also. So I mentioned that these patients are being put on the treatments, but how do you know that the response is working on those things? There are several technologies which are coming online which we can use. The T-cell repertoire. How is the T-cell changing, repertoire changing? Is it going in one direction, or is it increasing, or how the clonality is changing? And then I will not show you that examples. What's interesting, I will show examples of how we are using the cell-free DNA to monitor the response to therapeutic, and also how we can use the single-cell gene expression analysis to monitor the response to genomics. So this work was done by our recent acquisition, Sequenom, where they looked into the response, into the cell-free DNA to see the response of the immunotherapy. So this slide came from a 74-year-old patient who was diagnosed with squamous cell carcinoma, and they used the cell-free DNA to determine the copy number changes in the chromosomes. This patient was profiled practically literally every single month, and as you can see on the top, there are chromosomal copy number changes. It does not look normal chromosome. And as the patient went to the therapeutics, the chromosomal copy number variations became much more normal. Every single copy of the chromosome was expressed at the same level. And the technique is so sensitive that it can pick at the copy number changes at a very, very high resolution. So this is the other experiments where they did, where they were able to look at small, the question they asked was on the lower corner shows that is it a noise or is it a real copy number change? And when they looked at certain mutations like P53 in the tumor cells and from the plasma cells, the ratio of the mutation or tumor burden load was exactly similar to the copy number variations. That tells you that the technique and the sensitivity is there to be used both in the clinical trials as well as for clinical diagnostics. As I mentioned, that single cell sequencing not only tells you whether your clonality is changing, but it also helps us learn more about the biology of the tumors too. What we have recently done is use a 10x genomics platform where you can encapsulate the single cells inside a bead, and the Illumina libraries are prepared inside the bead itself. So each cell is tagged with a unique barcode. In a matter of days, you can profile about 60,000 cells, and based upon your gene expression profiles of these cells, you can predict how your patient is responding or which immunotherapeutic options are working or not. So in this particular case, we profiled about 8,000 cells just to see if the approach works or not. And just based upon the gene expression profiles, we were able to segregate them into different clusters. And now if your drug is working or to optimize your drug treatment options, you can determine if, for example, a green population is the immune cells, are they appearing or are they disappearing? Is their clonal population going down or not? Or what changes you need to do or combination therapeutics you need to develop so that these green cells of population can disappear. So in addition to the cell-free DNA, the tumor cells also shed the circulating tumor cells into the liquid plasma too. So in this case, we are working on the intensive trial for omics, use of omics in triple-negative breast cancer patients, where we have been longitudinally collecting both circulating tumor cells as well as the cell-free DNA. Now the question we are asking is there something which we missed by the whole genome analysis which can be used for therapeutic intervention in this case? And how the single-cell sequencing allows us to monitor this patient longitudinally or to give more highlights that this patient needs to be put on one therapy versus another therapy. And in the end, it's all about putting all these data sets together too. You have got the methylation data, you've got the genotyping data, you are looking into the gene expression data, how we can use the various computational tools to better understand which of these genes are expressed, when they are expressed, how they are expressed, so that we can identify change the biomarkers as the patients undergo treatment or not. These all tools are available for both use in the clinical trial settings as well as the diagnostics. And as a part of Covance, we have developed this web page where you can go look online what all resources are available for clinical trials and diagnostics. In addition to these resources, the global reach of companies like Covance allows the patients who are being treated in different countries or their samples to be analyzed appropriately. With this, I will pass this on to Adrian.
Thank you, Anup. So thank you both to Steve and Anup for those fantastic presentations. Steve did an exceptional job laying the foundation, and Anup followed up with a great in-depth review of how Covance is leveraging comprehensive genomic techniques in their work with clients. My role for this webinar is to moderate the Q&A section. However, before I do so, I wanted to call out a recent review paper that provides a proposal for personalized checkpoint therapy. In this paper, published in JAMA Oncology this year, the authors provide a framework for the future and how NGS could complement technologies like IHC for the selection of subjects for checkpoint therapy. In this figure, the authors call out 4 possible biomarkers that could be derived from either RNA or DNA sequencing. The first is the assessment of immune composition, and one could think of analyzing RNA-Seq data in order to understand the complex tumor microenvironment. The second is focused on tumor foreignness and immunogenicity, where DNA sequencing could provide information on mutational burden. The next topic focuses on how tumors can escape being targeted by the immune system. A potential mechanism for this could be found by using DNA sequencing to assess mutations in antigen-presenting machinery. And finally, the authors call out tumor extrinsic factors like germline polymorphisms or the composition of the microbiome, 2 markers that could also be assessed using NGS. This review paper does a great job of demonstrating the potential value that NGS can bring to anti-cancer immunotherapy diagnostics. And at Illumina, we're committed to helping our customers understand how NGS can be leveraged in their immuno-oncology research. With this goal in mind, we've created a website, and you can see the URL here. On this site, we have publication reviews, application notes, bioinformatic tech notes, and a listing of products that can be applied in the immuno-oncology space. And I encourage our attendees to check the site out. And so that brings us to our Q&A section of the webinar. So on slide 20, Anup, I believe this was a slide where you were talking about neoantigens and the tumor microenvironment. The question was what sample type, whether this was from fresh frozen or if this was from FFPE. And the other part of that question was, did you profile the tumor only or the microenvironment or both?
So these were FFP samples. The reason we wanted to do the FFP samples was that these are the samples available in the clinical trials. So all of the samples were clinical trials, and we profiled only the tumor samples in this case. Okay, thank you so much. I have a question here, wondering if a customer can follow up and see a tutorial or demo of the combined analysis by Covance. I'm assuming that's something that the Covance folks can get directly in touch with that person in order to do? Yes. Okay, and I'm not completely, I don't completely understand this question, but it's a very simple question. It's asking about the edge effect. What is the edge effect? So basically, the edge effect is something where the mutations are called or interspecially are present, and if it says, if I can go back to the slide, I can show you. The mutation is shifted a little bit. It's present in there, but because of the variant calling programs, if you have, let's say, a stretch of poly A tail, one of the or 2 of the bases are missing, instead of calling it the chromosome 1, 2 million, it calls that chromosome 200, like 5 base pair or 6 base pair shifted. That's what is called edge effect. And when you are comparing your now DNA and RNA-Seq data, the same mutation is present, but it is present at a little bit different chromosomal position. Okay, great. The next question is, given the sort of combinatorial explosion in immuno-oncology, what do you guys feel is the best biomarker strategy? And then as you develop biomarker sort of from a discovery process,
how do you anticipate those being moved into the development of companion diagnostics, just given the complexity of the biology and the interplay between the immune system, the tumor microenvironment, and the tumor?
Yeah, Adrian, that's a fantastic question. I think all folks working in this space are trying to use the appropriate tools that would allow them to look at both tumor microenvironment and other areas as have been discussed. I think with everything, with all areas in drug development, applying those earlier in the development cycle is relevant, and we're seeing that as even in simpler areas where folks targeted therapies, where individual companies are looking at potential companion diagnostics, incorporating the biomarkers earlier in the drug development cycle, working with partners that can build out assays that support trials, but also thinking about those under design control so that it can be migrated from the trial setting into a commercial setting. So early in development, right partners, right tools, I think are all the important things to really consider.
Great. How does tumor heterogeneity affect express mutations when compare exome with RNA-Seq?
Yeah, so see, that's the reason it affects the tumor heterogeneity is the biggest problem in all the fields, right? That's why sequencing it much deeper get rid of some of those problems. But again, some of the genes which are expressed at very low levels, or some of the mutations which are present in only a subpopulation of cells, those are not detected. And that's the reason the field of single-cell genomics is exploding, where you can look at each of the individual single cells, you can profile them and look into much deeper at the tumor heterogeneity, right? The approach I showed you with 10x genomics, it's not the perfect approach for doing that, because it just uses the 3' end of the sequences. A better way to do it is isolate those singles or those population of cells, and once those population of cells are isolated, and then today you can go down to like 10 picogram levels, where you can generate the sequencing libraries and do the whole RNA sequencing. So those approaches are available. The technology is there, where we can look at subpopulation of cells in a much higher depth, and then look at the tumor heterogeneity too.
Great.
I think
this is a question. Yeah, go ahead, please.
I would just add on to Adrian. Yeah, sorry, Adrian. I would just add on to that looking at multiple compartments, right? So biopsied material, blood, et cetera. I think as those again applications evolve, it will help address the challenge that we all face, where a single biopsy, a single core from a tumor sample, how representative of the tumor is that single sample.
Mm-hmm. This is a question actually that I've heard asked quite a bit to a variety of different folks, which is you end up on slide 19 with 125 mutations. How do you then prioritize and select the top several candidates?
Yeah, so there are various things which are available, right? You need to look at the net score, MSC scores are there. You look at various aspects of it. Sometimes it's very difficult to select the right one to go after, and you have to look what antibodies are available and how much time it will take. There are tools available online. If you go online, there you can do the search. There are a lot of tools available. There is no hard and fast rule which peptide is going to work better. You have to do the experiment in this case, and then select the candidates from there.
Great. What do you all believe are some of the gaps that really need to be addressed to improve the use of genomics in your research? Is this now an informatics issue? Is it an issue regarding sort of technical advances from the sequencing perspective? Are there a need for sort of larger clinical data sets that will help to improve and identify emerging biomarkers. Where do you see these gaps really existing today that will really continue to propel the use of these advanced genomic techniques, both in research and then consequently in the development of diagnostics?
I'll take a stab, and then if you want to follow up on that. I think the question actually identified all those critical areas, right? So I think having strong data sets, we've seen the value of the Cancer Genome Atlas project and others where we've looked at large data sets that have told us something about the biology of the tumor and maybe about the way in which we biologically look at that tumor type, not being one tumor type, but often being multiple tumor types within that group. I think as Anoop showed, the sequencing technologies, are getting tremendously better as we look at the biological problems that we're trying to address, whether it be RNA sequencing in expressed genes versus whole genome, whether it be single cells, whether it be circulating free DNA, right? So all of those we're seeing the evolution to match up with the sample type and the question to be asked and the problem to be solved. And then I think Anoop did a great job with the visual of how you pull all of that data together. And to me, I think that's still the biggest aspect, right? So we're getting good data out of larger data sets. The technology is evolving so we can do these very exquisite experiments, but then how do you pull it all together and make sense of it?
Yeah. Great. Another question here is, there's been quite a bit of work around sort of the deconvolution of bulk RNA-Seq into the tumor microenvironment. You, Anoop, showed a slide on single-cell technologies and their ability to sort of give you an idea of the tumor microenvironment as well via single cell. Which of these do you see as having sort of more applications in immuno-oncology research?
These are all different approaches. Again, going back, these are different tool sets which are available. You cannot pinpoint that one technology will give you the answer, right? You have to look from each tumor type, each patient, from variety of tools and variety of approaches. And the field is still evolving. The number of mostly immunotherapy is done for metastatic patients, right? And those patients, the population or statistically numbers are not there right now. And as we treat more patients, the more data we collect, we'll be able to say which tool, one tool is going to work better than other. It's still, as he mentioned earlier to the previous question, it's still a computationally challenging problem. We need to develop these tools where we can analyze this data set much more quicker as compared to taking months and months to analyze this data set, right?
Yeah.
Some of these patients have a very short time to live. They just cannot wait. And as those tools they get developed or those tools get refined, we'll be able to better answer the questions. If we can get all the information from single-cell genomics, perfect, great. That will be a great thing to do. But currently, we cannot escape doing the bulk RNA-Seq as well as the bulk exome-Seq. You still get a lot of peptides from there. You still get a lot of information from there. How do you prioritize those things? As again, we mentioned, like what rules we need to put in place that treat the patient, use one peptide versus another peptide too.
So with that, I'd like to pass the ball back or the closing remarks back to our moderator.
Thank you very much. Appreciate it. Thank you all for attending this Fierce Live webinar and submitting so many great questions. I'd like to thank our speakers for participating and Illumina and Covance for presenting today's webinar.